So many groups are there. So like this, so many groups has associated how the SIRT6 is working with the lifespan and all those things. So this presentation has a workflow in the way like how the palmitic acid has induced, like used for the insulin resistance as a model, SIRT6 histone deacetylase, and how the cellular dynamics varies with this, and what is the relationship between the SIRT6 and NRF2 target, and what are the different mitochondrial regulatory proteins which are interacts with the SIRT6, and at the end the conclusion is there. Next slide please. Next slide please.
Yeah. Okay.
So we have started this work like whether this SIRT6 involves in the insulin resistance in the palmitic acid model. So we started how the palmitic acid we have used. Next slide. So in this one, what we have done, we have given the 2 NBDG and we have established the relationship between the 2 NBDG and the control group, and we find out like how this insulin resistance has been established. Next slide please. Then we have given the palmitic acid and in 2 different combinations. Next slide. Where we have found that both the concentrations 200 and 500 microgram. So both this has leads to the insulin resistance. Next slide, please. Next slide. So then we have found out like what is the effect of this palmitic acid on the this mitochondria. Last slide. So in this last slide.
Yeah. Last. Okay. Sorry. This.
One more back.
Okay.
So in this slide, if you see when we give the palmitic acid here, and we will come to know that there is a more fragmentation as compared to the control. Next slide. So it means like palmitic acid is having some effect on the mitochondria. It is leading to more fission as compared to the fusion, which is very important for the working condition of the this cells or the mitochondria. So what is the effect of SIRT6 histone deacetylase on the palmitic acid induced this model? So in 2019, one of my paper, we have shown that there is a SIRT6 deficiency causes the glucose intolerance and impaired glucose stimulated insulin secretions. Similarly, many other groups has also shown the importance of the SIRT6 and how it affects the signaling on the development of the cardiovascular diseases, including the beta cells of the pancreas also. So just click on this, all the slide here, 2-3 clicks, you just done. So here what we have done, like we have used this 2 models where the SIRT6 overexpression model and SIRT6 knockout model, and we have given the palmitic acid. So when the palmitic acid we have given here, there is the insulin resistance as we have established earlier. But when we are doing the overexpression of the SIRT6, this effect of the palmitic acid is nullified by the overexpression of the SIRT. When there is a knockout, we didn't get much changes. But when we are giving palmitic acid in the knockout cells, we are finding that there is a more insulin resistance. So these cells, which are the knockout cells, are the more prone to the insulin resistance by the palmitic acid. Next slide, please. Just 2-3 clicks you did. So from here we come to SIRT6 overexpression overcomes the palmitic acid induced insulin resistance. Similarly, we have also looked for the different insulin resistance marker, which is the IRS1. We saw there is a more phosphorylation in the palmitic acid insulin resistance. The phosphorylation is more. However, with the SIRT6 overexpression, this phosphorylation is comparatively less as compared to the palmitic acid group. Next slide.
Should I keep on going?
Yeah. So here what we have, just next slide. Yeah. So here what we have done as compared to palmitic acid, where we have found that there is a more fragmentation in the palmitic acid. But when we are doing the SIRT6 overexpression along with the palmitic fusion as compared to fission, so mitochondria remains healthy as compared to the earlier one. Then we have also seen the different biomarkers of the mitochondria, and we have found that these biomarkers also varies with the this fission and fusions. So here in the mRNA levels we have seen, we have didn't find much changes in the SIRT6 mRNA levels, but we have found tremendous changes right after the 2 hours after giving the palmitic acid. Right after 2 hours, we have found that there is a protein expression gets changes with the SIRT6 right after 2 hours, and it is almost abolished by the 16 hours and 24 hours. Next slide. So SIRT6 is very, very important, and with the palmitic acid, it is reducing the expression of SIRT6. So then we try to find out what are the different targets which are may interact with the SIRT6. So we, as in the mitochondria, for the mitochondria, the NRF is very, very important. We have found that NRF2 is important, and we found that NRF2 levels are increasing with the SIRT6 overexpression, and the KEEP1 levels are reducing with the SIRT6 knockouts. Can you, yeah. So here we have seen that NRF2 mRNA levels and KEEP1 mRNA levels. So here we found that there is no change in the NRF2 levels. Here we have found that there are two in the, what are the changes in the fractions. So there is no change in the fractions of the cytoplasm. But if you see the nuclear fraction, there are NRF2 levels are very much reduced in the palmitic acid, but there is much more expression levels in the SIRT6 overexpressions. Then we have seen here the immunoprecipitation we have done, and we found that in the SIRT6 overexpression, the NRF2 levels are also very high. So as the SIRT6 levels are increases, NRF2 levels are also increased. That when there is a more SIRT6, there is a less KEEP. So KEEP is the antagonist to the NRF. It works oppositely to the NRF. So SIRT6 is more, KEEP1 is less, and we have found in the immunoprecipitation that if the SIRT6 is more, NRF2 levels are more. So there is a correlation between the SIRT6, NRF2, and KEEP1. So SIRT6 and NRF2 goes parallelly, and KEEP1 is going opposite to the levels of the NRF. So this is in the immunoprecipitation experiments, we found that SIRT6 and NRF2 go parallelly to each other's expression. However, the KEEP1 is works opposite to the NRF2 levels. So where KEEP1 levels are reduced, as we see in the figure B. Next, please.
Should I keep on going?
Yeah, just keep on. So
Should I keep on
keep
going?
going, please.
Okay.
Yes. Next one. So here what we have seen, see if there is a high SIRT6 levels, the KEEP1 levels are reduced. And if there is a less KEEP1 SIRT6 levels, then there is a more KEEP expression is there. Next slide. So these are the markers for the mitochondrial proteins, and these are HO1 protein, NQO1 protein, GST protein, and PRDS protein. And we have seen where there is the more SIRT6 overexpression, these protein levels are also increased. And when we are giving the palmitate, these expression of these proteins are reduced. So mitochondria is working directly proportional to the palmitate. When there is a more palmitic acid we are giving, these mitochondrial targets are reducing in the levels. So from this, we have found that, next slide. So we have established the scheme. In this scheme, just keep on going. Next slide. So if you see here, next slide, next click. Next click, please. So here what we have seen that if there is SIRT6 stabilization is there, then there is a NRF2 will come down. So here what we have seen that where there is a more SIRT6 expression, this SIRT6 get attached to the NRF2 and there is a and keep one remain away. And once the NRF2 stabilization is there is NRF2 regulating genes will get up. So this is how we can save the this cell health of this can be very good. Last slide, please.
This is conclusion slide. So do you want this?
Yeah. So this at the end, I can tell you like from this we have found that SIRT6 overexpression has reduced the palmitic acid induced toxicity. And SIRT6 overexpression induces mitochondrial fusion and hence leads to the mitochondrial biogenesis. SIRT6 binds to the NRF2 and contains the keep away, and this leads to the cell health by regulating the NRF2 regulating genes. Thank you. This is the overall presentation for today. And at the end, this is the picture of the AIMS campus in our Bathinda. Thank you.
Thank you so much, Dr. Kanwal. I think, yeah, should I stop sharing now?
Yes, thank you.
Yeah, thank you so much. That was a very nicely made presentation. I think so. The plan for the talk is that for the 15 minutes, we would have a brief research presentation that you have given. Next 5 minutes,
Right.
we would like to know more from you, like your journey after you reached India. Like, how did you decide on a topic? How did you grow your network? How did you decide a mentor? If you can give some ideas to our audience here who are aspiring to do the same, and then later
Right.
10 minutes we would take some Q&A from the audience. Yeah, so if you could
Okay.
tell us a bit more about your journey, like how did you start your group and how did you build a network?
Yeah. So right after my PhD, like I did my PhD from THSTI, Faridabad, and that is a DBT cluster. So then after that, I have been to the University of Chicago for my postdoc. So it was there in my mind, like I want to do postdoc at least for 3 to 5 years, and then if I get any good chance, then I'll come back. Why I come back is the reason, like I have seen many people who are during that time were trying to find the job back in India, and most of people who are staying like more than 10 years or 7 years, something like that, they're getting difficulty in finding the jobs in India also, and also in staying in India also, because the system they have fond of. So it's the life in US is like comparatively very easy and very good. So when I got the opportunity through the sciroi only, I attended the meeting in University of Chicago. Like every year, University of Chicago was hosted this sciroi. So there I got the opportunity in Manipal University, which is right now Mahe, and I came back in Kasturba Medical College there. And from there, I wrote Welcome Early Career Welcome Trust Fellowship. So that was also very good experience. I for the whole 1 year that process was there, and my application was short, like for the last round. And by the time it has happened, then I had given the interview in AIMS Bathinda, and IIT Hyderabad. So I got the opportunity in AIMS Bathinda.
Mhm.
So then I have joined AIMS Bathinda here,
Okay.
and the networking is like by the time when I came back in India, so few of the people I know in US as well as many people I came to know through the SCIRI also. And so we have developed this network through that and start writing the small projects together. So that's how we have wrote. And now like recently, like past 6 months, I got one grant from ICMR, which is in collaboration with nuclear medicine.
Okay.
And so this is also like what we have. That's how like it has been proceeded.
Okay. Okay. Yeah, that's really good. And then how did you start your group? Like, did you face any particular challenges in terms of facilities
So
or?
most important, yeah. So what I feel the most difficulty is to start to get the space of my own lab. That was I felt in the initial days. Starting a group, getting the right students, it's very difficult initially because if you get miss, you have no choice because there is a system of getting the uh, like entrance tests and all those things, right? So you will get a student from there, but the thing is space is very important. The space is very important. For that space you need some funds from outside, like extra mural funds. If you are having, then the definitely that organization will give you space. So that is the challenge in the initial days. And then I have started my group with two like projects with the two students, which are the like postgraduate projects, like for one year. And after that I have got one student where she I wrote a one grant for her for her fellowship. And so we both together have did work and she got that grant, and she is now the PhD student of mine. And then also like I am a co-PI of two students, so that's how the group has been built now.
Okay.
So yeah, that is the way like how the things has been proceeded.
Okay, okay. Yeah, sounds really good. And thank you for sharing all of that information. I think now we can let our audience or some participants, if you have any questions, you can raise your hand and then I can call out your name. Any questions? Yeah, if there are any questions about, yeah, please go ahead,
Hi,
doctor.
sorry, I didn't see the option to raise hand, so I'm just asking. I'll just share my video. So I'm Dr. Manu Smithi. I'm at Bennett University in Greater Noida, and I did my PhD from Germany and postdoc from Israel Tel Aviv University. So it's been a year. I'm here, and for me the problem is I my work is on nanomedicine. I prepare nanoparticles. We've done siRNA delivery as well as drug delivery before. So since my PhD and postdoc, I've been abroad. I am not familiar with the research system in India, and I am finding a little difficult to build a network. And my work is very clinically oriented. I would wish to have
Right.
clinical samples. So what is your suggestion? I tried to speak to some of the hospitals but it has not been very fruitful. So if you can give your insight.
Yeah, so the thing is like sometimes initially it's very difficult to approach the people here in India. And because the thing is uh, especially in the clinical side, like if you are not the part of that clinical side or we are not making any network with them. And even I have seen that there is some hesitant like resistance from the clinicians, like to give the samples and all those things to the other people. So like from my side I can tell you like I work in the aims. So means from my side, if what I can do best for you is like we can collaborate here also, and the samples we test, okay. uh, but what I can tell you is like making the networking is here is like either another important thing is like when you came the COVID has started. So So that is another important thing, because you cannot travel from one place to another place very frequently, or some conferences were, most of the conferences were like online, right? So, and so even like another thing is like the funding agency has stopped giving the fundings in between. Like I got my ECMR project like after 1 year delay. So that is another thing. Now the things has been much better, and I hope like you will get better responses from other people. But if you need any type of help from my side, like you can just approach me. I don't mind. Like I will be very happy to do that. So yeah, so that is like how we can. I
Yeah,
can
my
share
work
my
is
email
on,
ID here.
yeah, can you please just share it on the in the chat
I
box?
will
I'll
share
share
in
it.
the chat box.
Yeah, my work is on cancer, but it's nanomedicine, so we can extend and we can think about potential projects. So I do have one seed grant from university around 30 lakhs and another seed grant copy I have written to SERB for a potential project with IIT. So let's see. I'm hoping that I can build a lab here as well.
the only the only thing what I have seen here is like when we come back from abroad, it is very important to have a patience. Actually, that is very very important. So please have patience.
Thank you. That is actually a very
Yeah,
useful advice.
Bec-
Yeah,
because
in this
sometimes we
corona.
get sometimes we get nervous, like the things are not going well, we should go back, or the opportunities in the abroad were much better. But I think like just hold on and just keep doing.
Thank you, Dr. Abhinav. I'll write to you. Yeah, thank you.
Thank you.
Thank you so much, Dr. Abhinav. And any more questions we have from the participants? So if not, we would move on to our second speaker because we're already like 6 minutes behind the schedule. So we have Dr. Ketanjali Chawla. And yeah, Dr. Ketanjali Chawla, so she is a welcome DBT Intermediate Fellow at RCB. And I think Dr. Chawla, yeah, we welcome you, and the floor is all yours. So you can start with your research presentation, and then we can ask you more about your journey.
Okay. I'll just share my screen.
Sure, sure.
Can you hear me?
Yes, we can hear you properly.
Okay. So I actually need to reduce the size of the, okay. I can't read my, okay, here I am. Fine. So good evening, everyone. And as she introduced, I'm Ketanjali, and I'm DBT Wellcome Trust India Alliance Intermediate Fellow in the Regional Center for Biotechnology at Faridabad. And today I'll be talking about how diet and non-coding RNA networks regulate healthy lifespan. So the clinical, it's not going to the next slide. Okay, so the clinical problem that my research program is addressing is aging. Aging is the process of getting old, or it's the accumulation of changes that occur in an individual over time. And though there have been a significant improvement in living conditions as well as better medical facilities, and that has resulted in a tremendous increase in life expectancy. So and it has been predicted that by 2050, the number of individuals over the age of 60 will be 5 times of that in 1950. And if we specifically look at India, in 2015, India, 7%, less than 7% of India's population was age 65 and over. And it is predicted that by 2050, 14 to 21% of the population in India will be aged 65 and over. So as the number of older people increase, so will the problems associated with aging. So aging is a major risk factor for a number of chronic diseases, including diabetes, cancer, Alzheimer's disease, cardiac disease, Parkinson's disease. So one of the major essential goals of researchers who are interested in the biological aspects or biomedical aspects of aging is to find a solution to this impending impact of aging. So, and one strategy that was proposed more than 40 years ago by a scientist named Fries is the compression of morbidity. So based upon this strategy, if we can figure out a way to delay the onset of these diseases to a much later part of life, and this delay is greater than any kind of increase in life expectancy, we can restrict these diseases to a much shorter duration at the end of life. And one way that has been proposed that this can be done is to slow down the fundamental biological process of aging. So if we understand what are the molecular mechanisms underlying aging, and we can design different interventions to slow down this process, we can delay these diseases associated with age to a much later part of life, in the life. So basically increasing healthy lifespan or healthy life expectancy is the strategy. And towards that goal, there are several groups that have focused using different model systems to basically understand what are the molecular pathways that are regulated during aging. And what they have found is that aging is surprisingly plastic. It can be manipulated. The rate of aging can be changed by dietary interventions or by genetic interventions or by pharmacological interventions. And so some of the models that are used in aging research include Saccharomyces cerevisiae, which is a single cell eukaryote, C. elegans, Drosophila or fruit flies, and a mouse model. And my lab is using Drosophila melanogaster as a model system to study aging. And some of the advantages of using this model system is that it has a short lifespan of 60 to 90 days. It is relatively inexpensive to handle compared to, for say, mouse models. It can be grown in large populations in the lab in vials, glass bottles, and in cages. And we can do population experiments that are essential for measuring lifespan. There are several tools available in Drosophila model system. We can easily change the expression of genes. We can modulate the dose of genes to look at genetic interventions. And then longevity pathways that have been identified in Drosophila are conserved in humans and can be manipulated in similar ways. So research in these different organisms has identified one intervention that can reproducibly enhance lifespan in diverse species. And this intervention is dietary restriction. Dietary restriction is reduced food intake without incurring malnutrition. And reducing food intake has been shown to extend lifespan in single cell yeast, in worms, in Drosophila, in rodents, in non-human primates. And in humans, dietary restriction has been shown to improve metabolic health parameters. And more specifically, if we, can you hear me? Am I audible?
Yeah, I can hear you.
Oh. Okay, so several dietary restriction has also been shown to be beneficial in different disease contexts. So disease models, if you can do different kinds of dietary restriction like protein restriction, amino acid restriction, there is branch chain amino acid restriction, methionine restriction, so different intermittent fasting. So these can all improve disease parameters. So you can, for instance, in diabetes, you can there is an increase in insulin sensitivity, decrease hyperglycemia, and reduce obesity in mouse models where dietary restriction has been prescribed. But however, it's not always beneficial because reducing food intake can also lead to a reduction in bone mineral density. And so it is not recommended for osteoporosis and also in certain cancers. So basically coming to how does it work? So mechanisms underlying dietary restriction are complex. There is no single pathway that can account for all the beneficial effects of reducing dietary intake. But this is a very simplified model to show the metabolic reprogramming that occurs with dietary restriction. So reducing food intake results in changes in signaling molecules, which are reflective of the nutrient status or the energetic status of the cells, tissues, or organs or in an organism. And the changes in the levels of these signals are actively recognized by very sensitive effector molecules. And these effector molecules, together with circulating growth factors, then lead to a change in metabolism, growth, in how energy is used in our system, and also energy sparing. And together, these lead to changes in the rate of aging. However, it is not clear how these changes lead to slowing down the rate of aging, and this is still under investigation. So my lab is focused on trying to understand what are the post-transcriptional mechanisms that regulate aging and dietary restriction. And the class of post-transcriptional regulators that we're looking at are microRNAs. So microRNAs are a class of small non-coding RNAs that range in size from 19 to 24 nucleotides, and they function by directing effector complexes to target mRNAs. And microRNA ribonucleoprotein complex interacts with a target through imperfect base pairing interactions between the microRNA and its target. And this results in either degradation of the target mRNA or translational inhibition of the target mRNA. So microRNAs generally or predominantly reduce gene expression. So in my lab, we are using Drosophila as a model system to test the efficacy of microRNAs in promoting healthy lifespan. And towards that goal, initially, I first identified microRNAs that are regulated by dietary restriction. For that, I exposed fruit flies to either ad libitum or high nutrient diet and a DR diet, which in fruit flies, it can be done by reducing the protein content in the diet. And we isolated RNA and then performed a small RNA-seq. And this analysis led to identification of some conserved microRNAs that are regulated by age and dietary restriction. And my lab is currently trying to test whether modulating these microRNAs can enhance lifespan. And those microRNAs that enhance lifespan, we are looking at metabolic or biochemical parameters to identify what are the metabolic pathways that they regulate. And then we're also using molecular approaches to identify what are the downstream targets of these microRNAs. So we are using proteomics, metabolomics, molecular and genetic approaches to test the efficacy of microRNAs to function as mimics of dietary restriction. So that normally individuals or organisms that are fed normal diet, if we change the microRNA level, can we see an enhancement in health? So one of the genes that my group recently worked on and analyzed is the LET7 complex locus in fruit flies. It encodes 3 ancient and highly conserved microRNAs, MIR100, LET7, and MIR125. And we initially found by small RNA-seq that LET7 and MIR125 levels increase with dietary restriction. So, we've recently published a story in eLife, and these are the 2 lab members who did most of the work. Manish was the lead author, and Sakshi Bansal helped him. So basically, in fruit flies, I've just put some data from that story to give you an idea of what are the assays that we are performing. So this is a lifespan assay, and it is a well-established protocol dietary regimen of by reducing food in protein amount in the diet, which is shown here in blue. Fruit flies also live longer compared to fruit flies that are fed a high protein diet, which is ad libitum, shown here in red. So first we validated the small RNA-seq data, and we found that the levels of the LET7 and MIR125 increase upon dietary restriction. And so does the level of MIR100, which is co-transcribed with these 2 microRNAs. Then we've also gone on to, furthermore, we found that genetic loss of function mutations in the LET7 microRNA or the MIR125 microRNA led to loss of the dietary restriction dependent increase in lifespan that we see in the control flies. So implying that these 2 microRNAs are required for dietary restriction mediated lifespan extension. And we also found that this phenotype in MIR125 loss of function mutants was due to elevated levels of its target known as KINMO. So KINMO is chronologically inappropriate morphogenesis. It is a transcription factor, and its level increase in the brain in MIR125 mutants. And this increase leads to deregulation or in fat metabolism, which I'll just show you. But if you reduce the level of KINMO in these mutants, by RNA interference or by genetic loss of function mutation, we can restore or rescue the mutant phenotype. You can see that these mutants then have a lifespan increase upon DR. So suggesting that MIR125 regulates this target KINMO, and that is what is required for DR-dependent lifespan extension. So these are some of the assays that we did. We looked at lipid metabolism because it was already published that when protein amount is reduced in the diet, this leads to a higher lipid metabolism turnover rate, or lipid turnover rate increases. So one way to look at it is by biochemical assay, or try by measuring the stored lipid levels by TAG assays, which I have not shown here, which is in the paper, but I'm just showing. We can also stain the fat body or fat tissue of flies under AL. The flies that are fed the AL diet have smaller lipid droplet size, and upon DR, the lipid droplet increases. In the mutant, you can see the lipid droplet size is much lower. So lipid metabolism is depleted or less in MIR125 mutant. But in this mutant, if we also reduce kinmo levels, we can rescue some of the phenotypes. So either by RNAi, there is some rescue, and if we genetically mutate one of the kinmo loci, we actually get even better rescue. It becomes like wild type. So what our data showed, to cut the story short, is that kinmo is a relevant target of MIR125. It is regulated not only by MIR125. So just to show you, in wild type flies, MIR125 levels are high in the central nervous system. So kinmo is completely repressed, so no staining. So basically you can't see any signal here. But in MIR125 mutants, the repression is lost. Kinmo levels increase dramatically in the brain and also in the fat tissue, which you can see the staining here. But kinmo is also regulated by diet. So if you move wild type flies or control flies to dietary restriction, if you expose them to dietary restriction, kinmo levels increase in the fat tissue. And this increase in kinmo levels is responsible for targeting fat metabolism genes. So we performed proteomics and in flies that overexpress the gene, and we found the different genes that are regulated by in the fat metabolism pathway. So basically what our story was that dietary restriction leads to lifespan extension by regulating the levels of a microRNA. MicroRNA is responsible for knocking down expression of its target known as kinmo. But kinmo in the brain is also non-autonomously regulated in the fat tissue, which I didn't have time to show the data for. But dietary restriction also regulates kinmo by post-translational modification. So kinmo is also downstream, is regulated by SIR2. And we just heard a talk about SIRT6. So SIRT1 is SIR2 in flies, and basically it deacetylates kinmo, and this deacetylation of kinmo moves it from the nucleus to the cytoplasm, and relieves repression of fat metabolism genes that leads to increased lifespan. So this is one of the main projects. We are currently analyzing other microRNAs which were identified. And just to summarize what I've just told you, my lab uses fruit fly model system, and we are trying to test the efficacy of microRNAs to promote healthy lifespan. So our goal is to identify those microRNAs that can fine-tune conserved pathways and can be used for broad spectrum health improvement. We also hope that some of these microRNAs can be used as a cost-effective strategy to treat multiple age-related diseases simultaneously. So another side project which is related to this one is to identify or evaluate natural and biofortified dietary interventions using the fruit fly model. So medicinal plants have been used for decades to improve health. So many of us have these natural interventions, but molecular pathways that are responsible for these beneficial effects have not been understood or are not known. And more specifically, RNA-mediated pathways involved in the beneficial effects of natural medicinal plants or biofortified plants are not explored extensively. So we are actually screening a lot of these natural available medicinal extracts to find out which ones increase lifespan. Then we are also testing whether they can increase health span by exposing fruit flies to oxidative stress, and then testing whether changing their diet using these interventions can improve their ability to survive in these under harsh conditions, in high fat diet or in oxidative stress induced conditions. So one of the stories that my lab recently published in Experimental Gerontology is looked at, we evaluated biofortified wheats that were generated by scientists in the National Agricultural Biotechnology Institute in Mohali. So biofortification is a way to increase the nutritional quality of normal crops. So in India, wheat is very predominantly used in the diet. So what Dr. Monica Gard's group generated these colored wheat variety lines by crossing methods, by genetically crossing them to other lines from Japan. And so these lines, the important thing is that these are colored because of phytochemicals known as anthocyanins. And anthocyanins are rich antioxidants, have antioxidant properties. So one of the key theories of aging is the free radical theory of aging, that proposes that aging occurs because of increasing oxidative damage that occurs in the cells and tissues. And so one of the main area of research has been to test whether antioxidants can increase lifespan, and the results have been variable, and increasing just the dose of vitamin C or vitamin A or beta carotene does not help in diseases, in heart disease or in cancer. So because most of these vitamins, they work in combination. So if you eat a plant-based diet which has antioxidant, which has other micronutrients, it is more beneficial, and there are enough clinical trials to suggest that. So what we did is we tested these wheat varieties which have increasing levels of anthocyanin contents or antioxidant amount, and we used our assays that I described. So we tested whether feeding flies colored wheat diet, can it increase lifespan of flies? So we found that yes, blue wheat, one of the, which is not the highest, which does not contain the highest content, but the second highest anthocyanin containing wheat, it prolongs lifespan under different conditions, under high protein diet, under low protein diet, and also under normal diet. And then we also tested whether when we expose flies to oxidative stress, can these wheat-based diets improve the survivability of flies? And we find that blue wheat supplemented diet is able to increase the survivability. So it is beneficial both in normal dietary condition, under oxidative stress condition. And then we looked at cardiac structural pathologies, which we can see in flies which are fed a high fat diet. So cardiovascular diseases are linked to obesity and high fat diet. So we can do those experiments in flies, and then we expose them to high fat diet, and then we test whether blue wheat supplemented high fat diet has a better profile. And we find that the heart structural deformities are reduced when we have these anthocyanin containing wheats. So these are the kind of experiments that we are doing in the lab. And this is just to provide a flair. I didn't put much data, but we have these 2 stories published in the last 4 years. And the topic that I want to talk about is that has been most positive for me is the negotiations for lab space and remodeling of the lab. So when I joined, I inherited a lab of Dr. Dinkar Solanki. He's a structural biologist, and my work required me to have a fly room in the lab. So this is how I first made a blueprint of the existing lab and tried to remodel one corner of the lab to make a fly room. And so this is just an inset showing that this was a small area in the lab that was being used for computer modeling stuff, because it was a structural lab. And then I basically changed it to a microscopy room or a fly room, which has to be enclosed. And so we made glass walls in the lab, and we had a lower benches to have my 5 microscopes in the lab, then electrical outputs, fly incubator. So these are supposed to be in an enclosed area. And my institutional administration was extremely positive. They approved the model. And this is how the lab looks like now. So this is the glass wall you can see. Here is a sliding door. We enter the fly room here. These are 2 benches, which is here, 2 microscopes here. And on the side, I have 2 more microscopes here. So these shelves and the electrical fittings, microscope, everything was done after I joined, and it was pretty fast, and the engineering department helped a lot. So you have to figure out how to get the approvals, and then it can be done. Then another room that I remodeled was my cell culture. I needed to do fly cell culture. And so I actually was approved to use a room which was originally used for storing furniture. So we had to basically include a sink, air filters. To make it a cell culture room, you need to have clean air, and then double 2 doors. And then I designed this kind of blueprint to have so that 4 biosafety incubators could be included in that. And then this corner belongs to my lab. So this is how the room looks like. So the sink and the storage space, biosafety incubators, everything had to be done. So the room is not only beneficial for my research program, this is my area, but now 5 other faculty are using that, and there are 25 users for this room. So it has been a positive experience. So I wanted to talk about that. So overall, my research career or the past 4 years and 2 months have been very positive in terms of research. So my fellowship allowed me to not only launch my independent research program, it helped me to set up my lab focused on Drosophila microarray biology. It helped me to establish. So India Alliance is pretty, it helps you, they have these conferences and meetings and you get to talk to people, other people working on different topics. So I recently established a collaboration with a public health early career fellow in Kerala. It wouldn't have been possible if I didn't have my, if we didn't have the India Alliance annual meetings. So, and then I already had a mentor and several researchers, some of them which I know already from before. So I'm extremely grateful for the fellowship award because it helped a lot. These are, and nothing can be done with efficient and nice lab members. So I had a very small team too, but these lab members have been extremely efficient and supportive. So this is my current lab. I have 2 trainees and 2 project JRFs, and this is a lab attendant provided by the institute. This is the Regional Center for Biotechnology. I'm also grateful to the Fly Stock Center. World Fly Stock Center is in Bloomington, Indiana, and the RCB instrumentation facility, and my funding sources. And I thank you all, whoever are attending, and I'm happy to take any questions.
Thank you so much, Dr. Chawla. That was a very, very nice, very clear presentation. And you answered everything that I wanted to ask you. So thank you so much. I think we can just move on to questions now. If there are some questions from the audience, you can either raise your hand or you can unmute yourself or speak up, or you can post it in the chat also.
No questions?
They understood everything, that's why.
No, okay.
We can wait for 1 more minute maybe. Any more questions?
Okay. That was easy.
I can ask one. Sorry.
Okay.
This is Vikas here. I have a question regarding your fellowship timeline and how intensive it was, like some experience like it.
It's very competitive, I can tell you that. And it gets harder as you move from preliminary to the second stage. And the things are very specific. You need to know what you're talking about. And the best thing is to focus. So fellowship is different from grant application in the sense they see how a fellow, how good the scientist fits into the project. So you need to have you have to give a very strong case of why you are the perfect person or unique individual to carry out your project. The project has to be good. It has to be extremely well written. And the hardest part is the interview. So in the interview only, there are very few people are called for interview, and then 50% chance. So basically you can you're out or in. So, and it's really tough. So hardest was the interview, but the process, the people, you get assigned a grant advisor or whoever, someone manages, so you can answer, you can get all the doubts answered. So it was very streamlined. It's very organized. And even after the funding, they are very, you can use your funds, flexible funds. You can move your funds from one subheading to the other. India Alliance is extremely supportive. It's the best. And everyone should try it if they want to come back, because it's one of the most prestigious. I also got the Ramalinga Swamy award, but India Alliance has a dual fellowship policy. So you can only hold one fellowship. And it has more funds. So I chose the India Alliance. But it is good. It is a good exercise to apply, but you need to know your project in and out. You need to be the perfect person for the job, and it is competitive. It's very high, and many people apply, and it's competitive.
And what about the preliminary data? Like how much do you need? And because if you have decided, if you have already decided,
I did have a preliminary data, so I think it will help. Because it will show that it is your strength, what you are good at. So it is good to have preliminary. I'm talking about intermediate fellowship. So I included preliminary data. I included some unpublished data, which I was going to use. And I think it only strengthened. Yeah.
All right. So the institute, if it is new institute, you're planning to join, like if you're not joined, so how that is like difficult in that, like if you haven't joined the institute and you were proposing something.
I did that. No,
Okay.
yeah. So I had applied. I had an associate professor position in Amity University. You're talking about whether I switch institutes.
Yes. So
no.
you applied for the institute where you are applying that this will be my host
But
institute.
I didn't join. So I switched the institute to RCB before I joined, after getting the fellowship. So it is hard. So don't do that. My recommendation is, if you have a position somewhere, please go and see the institute, see what are the facilities. It actually, you lose friends, and it is overall, if I have to do it again, I would choose the institute and stick with it. But there were other considerations in my case, which I cannot discuss. But if I have to recommend anyone, if you are getting the fellowship, then you join the institute, or you choose the institute very carefully. But a Welcome DBT gives you the option to change your host. But it depends on how strong you are. It's hard. I'm telling you that. So try to avoid if possible. But fellowship is a good thing. You should apply. Anyone who's eligible, I think it's a very good fellowship.
And how many pages like does it document?
Well, the pages, you have word limit everywhere. So it's a very different application. So you get assigned basically you have a space and you keep adding. There are a lot of documents that you have to fill. Don't do it in a hurry, because you will not get it. You have to read it, reread it, improve the quality. It helped a lot for me to, you have to sleep over it. You have to wait, then read again, you'll find more mistakes. The more mistakes, the lesser the chance. It won't move to the next step. And you get only one chance. Basically, you cannot apply for the same fellowship again. So do it well, take your time, think about each and everything. So I told you that 50% is about you, how you write about yourself. So how your personality and how you fit the project, what your training has been, and what are your strengths. So focus on those. Right, you have to write about all this and the project. So how you fit with the project is very important for them. The project is definitely should be novel and new, and should be nowadays it's related to any biomedical. If you're applying for the biomedical one, look at problems in India and whether your research can help that. So write about unique problems, and then write about how unique you are to solve those problems, how your training has been to make you unique.
Okay, thank you.
Thank you so much, Dr. Chawla, and thank you, Dr. Vikas Verma, for your questions. I think in the interest of time, we would like to move on to the next up. Again, thank you so much, Dr. Chawla.
Thank you.
So next we have, next we have Dr. Syed Ilyas Basha, who is an assistant professor in pharmacology department at Geetham Institute of Medical Sciences and Research. So Dr. Basha, if you would like to switch on your video and start sharing your slides.
I am audible.
Yeah, you are audible. Yeah.
Good evening, one and all. That was a fantastic presentation by the Dr. Chawla. Share the screen. My end.
Yeah, please go ahead. Okay, we can see your slides. Yeah.
Yeah, thank you. So this is my work, which I recently published. Basically, working as a assistant professor in Medical College, and very new to the research field. Hope you are aware the kind of PhDs and this thing will in medical colleges. So So we have developed a silver nanoparticles hydro formulations, keeping in view on the diabetic wounds where there is a problems of like aging and metabolic disorders, formation of biofilms and prolonged inflammatory phase, which again triggers the oxidative stress or the reactive oxygen species. That is a central area where in the diabetic wounds leads to the multiple factors which can affect the diabetic wounds and hamper the wound healing. In India, basically, you are aware that majority of the population, if in a family, at least 2 or 3 are diabetes and hypertension, suffering from these two metabolic disorders. Particularly if you take the wounds and the diabetes, it is a very huge burden with regard to the health management and with regard to the financial burden in managing the diabetic wounds. So what a formulation should be, what we desired, it is the features of an ideal wound healing agent. It should be cost effective, or it should be act as a barrier to the external contaminants, provides proper hydration, because hydration itself has giving a lot of angiogenesis in the area of wounds. It should act as an antioxidant and better absorption and antibacterial property, biodegradable and coolant effect. It's all are the characteristic features which we design in one formulation, keeping in with regard to the diabetic wounds. So is the roadmap where we have first we synthesized the new drug molecule, incorporation of a new drug molecule and synthesis of agar-based nanoparticles. Then we have characterized it. Then as a proof of concept, we have tested the formulation for its biological activities like antimicrobial and cytotoxicity studies, where later on after getting the successful in vitro studies, then we have moved to the in vivo, where we have tested the formulation for its wound healing activity, both on diabetic and non-diabetic wounds. So this is the process how we have developed and synthesized the nano formulations in 3 stages. I think you can find out in my paper. I'm not going to describe this in detail. So the lead compound, how we have synthesized the hydrogel, then we have characterized through UV and scanning electron microscopy and Fourier transmission, and we have tested its degradability and silver releasing properties. Then we have developed the standardized the formulation. These are the results where you can see here. These are the multiple formulations we have developed. One is what we have done is we have incorporated the silver nanoparticles in simple agar-agar, which is a nutrient for the microbes. So here the agar-agar will act as a trap for the bacteria as it engulfs, and the silver nanoparticles and the active molecules will kill the bacteria and hamper the biofilm formation. So when we have incorporated not only the silver, that is Ag, and AgFA and EFA, we have incorporated, we have tested the stability of the compound. The 450 nanometers describes the formation of the silver nanoparticles even after the incorporation of the other active components in the silver. That means silver has not reacted with the other active compounds. The same we have tested with the same molecule, the sister compound of this, where we found the same results, but its kinetics are different when compare with the A compound. This is the characteristic, the size where the 12 nanometers, size of the silver nanoparticles. And the SEM images, which describes the morphology of the formulation. And this proves the antibacterial property compared with the standards, where the synergistic activities has been shown by the formulation. We can see the 4 and 4 here, all the E. coli or Pseudomonas stuff, which are the culprits in the diabetic wounds, particularly the Pseudomonas and Staphylococcus. And we have tested for its cytotoxicity studies, where these images revealing that the compound is not that much cytotoxic, where we have we can see the live cells in the green images and the dead cells with the red dots. That is the AOAV staining. And further, we have tested for the in vitro cell proliferative assays, where we confirm the formulation has the property to proliferate the cells in vitro, which confirms the compound having the wound healing property. Maybe it gives a faster wound contraction rate that we have done under 0, 12, and 24 hours. So you can see here in higher concentrations, the proliferation is much better than the control. So then we have tested the formulation on the diabetic wounds, where you can see this is the diabetic excision wound, where the compounds AA, AGFA, and AAGMA showing the better, much faster contraction compared with the negative control and the standard mega heal gel. This is on day 16 and day 18. Not only the physical appearance of the wound, but the physiological changes and as well as the quality of the wound has been characterized, tested under the histopathology through histopathology studies. This is the article you can find. So basically, my interest in this talk is I'm searching for the tie-ups or the associations where I can bring the compound for the develop as a clinical trials area. Believe me, that this compound is giving good results, where many patients with diabetic foot ulcers can be treated within no time. Even the most damaged conditions or the foot or the parts of the body can be treated with this compound. Thank you.
Thank you so much, Dr. Basha, for your nice presentation. So for the next 5 minutes, maybe if you can tell us a bit more about your journey, or is there any specific topic you would like to address? Like how was your journey? How did you grow your network, your research?
Yeah, basically, journey was very tough for me because my study was a multidisciplinary study where I have to work in the field of chemistry, microbiology, and other characterization studies. I have to do a lot of struggles I have to go through. It was not that much easy, but somehow I thank my mentor, Dr. Somnath Ghosh. He was a working as a dean in Indian Institute of Petroleum and Energy, Visakhapatnam, Andhra Pradesh. So somehow he gave me the contacts to maximum extent. I got the help from the IISc Bangalore and some other Chennai-based commercial labs by spending money like that. So it was very tough. It's very, I can't explain. deeply.
And how is your journey as an assistant professor so far?
Yeah, in academics, it's no issue because what we do is nothing. Just we take our classes, we come back. But when come to the application of the subject in research, and we need a lot of collaborations. That is a biggest challenge, particularly in the medical colleges, where the medical fraternity may be not all, but majority of the people won't comfortable with the collaborative type of research in medical colleges. Since my background is medical MSc pharmacology and PhD, so it's difficult to make a collaborations with the people in doing my study in clinical trials and like that. So then I'm seeking help from the external resources. If anybody interested to take the compound and to work, so as I can work with them to developing the compound.
I hope through Sci-ROI India's this event you can find some more resources and more collaborations that you can make for your research. That would be helpful. And thank you once again for your talk. I think now if there are some questions from the participants for Dr. Basha.
I do have a question, if I may.
Yeah, please go ahead, Doctor.
Yeah. Thank you. Very nice talk and very good positive results, Dr. Basha. I wanted to ask if you've used any positive control because there are some commercial gels, for example, SilverX. It does have silver and it's a gel-based cream which is used for inflammation on skin or something. So did you use any positive control?
That was the Mega Hill was the positive control, which I showed in my slides.
Okay. Thank you very much.
Okay. Any more questions? Or or if there is any other question for Dr. Chawla or Dr. Kanwal. So I think if not, then maybe we can end this session if I'm not hearing any more questions. And I would like to again thank you, Dr. Basha, Dr. Chawla, and Dr. Kanwal very much for your very nice presentations and for taking out your time on Friday evening. So thank you so much. And I hope you enjoy the rest of the event as well.